been able to make because everything meshes together so nicely. So I want to say thank you very much for the invitation, and it's pretty exciting to be here. And I want to admit that I wasn't familiar with the words embodied intelligence before I was invited to do this, and I've had so much fun thinking about my work in that context with that new title, and hopefully that comes across today. Before slide, there we go. Before I continue on too much, I would like to uh, just do a shout out to people that have brought me to this place. So collaboration is where science grows, and for sure, um, I have to acknowledge Akio Ishiguro and Aoka Ishbert, who have brought me into the world of biorobotics and simulation, and have just opened my mind to how cool everything can be when we have tools that we can break things apart. I'm very much a biologist and deal with evolutionary biology, and I think about movement from a biological context. And the tools that robotics give us to sort of take apart the complexity of biology are really exciting to me. Okay, so I like to think about locomotion with sort of three different fields, if it were. So there's the external mechanics, the internal mechanics, and the neurobiology side of things. And so um, the world that animals move in matters. And I was very excited about the phrase ghost circuits because I think about these all the time. Sort of the combination of external and internal, how the internal of the animal is operating with the external environment and how that results in performance and locomotion. And then, of course, neurobiology is critically important because that is the network of commands that can be given. And so to start this talk off, I wanted to talk about the neurobiology of things. And of course, to talk about neurobiology, uh, we start with mice. I don't work on mice. I work on things that swim and crawl, but mice are an incredible model. They're tractable. There are many mutated strains, and so you can use tools like optogenetics to turn neurons on, turn neurons off. And when you're thinking about the complexity of neurobiology, these are really fabulous tools to understand which parts are playing a role. Okay, so thinking about the mouse. Very easily, we've got a brain and a spinal cord. These things are command central for locomotion. We all know this. They create motor output. The other thing that's critically important is the sensory feedback or the sensory input that animals feel. And there are two kinds of sensory input. The sensory feedback that comes from vision, I call it sort of top down, vision, smell, hearing. These are all things that filter through the brain. The brain passes a signal to the spinal cord creates a motor output, and then you get your second type of sensory feedback. And that's feedback that can go directly back to the spinal cord to alter the immediate motor control of the circuit, or go back to the brain for some computation, which also can alter the circuit. Now, in the sort of neurological part of embodied intelligence, it is very much that spine that I am interested in thinking about. Oops, now we're working. Okay, so if you look at what people know about the way circuits work, this is very straightforward, and we've talked about this in many talks this morning, is that you've got feedback and feed forward. So intention of an animal goes to a controller. That controller spits out a motor command. We call that an effector. Let's see. If I do this, then you can see what I'm pointing at. We call that an effector. That effector has sensors associated with it that sends a message back to some sort of cell, interneurons or the like, and can send the message back to the controller to alter the mechanics. Now, feed forward is simply exactly the same motor command, but an efference copy of it going to a cell. And if you have the sensor feedback and the feed forward, predictive feed forward, coming into the same cell, what you have is a comparator system that can then create prediction error that it passes to the controller. And this is a really precise way to make small and effective changes to your locomotor pattern for highly effective locomotion. It's also an excellent way to establish learning. And so it's well understood that this occurs in the brain. Cerebellum is busy with comparator circuits that people have identified. The big question is it happening in the spinal cord? And so with the idea of neurobiological embodied intelligence, it's really the spinal cord that is where I'm interested in thinking what sort of computation or intelligence exists there. Now, if we look at this, here's a diagram. This is essentially the same diagram that I showed you before. It's out of Brownstone, Bowie, and Stefani, which is a great paper if you're interested in understanding how biologists think about these neural circuits. Here are your effector muscles. They send a sensory signal in. That sensory signal, when intact, can go up to the brain, to the cerebellum to do some learning. It can also come down to the comparator cell, which talks to the controller that sends the efferents copy to the comparator at the same time they're controlling the muscle. So you've got this circuit in here, but you can see this dark gray line coming in is the intention. So this is the brain sending the signal for what to do. The question really truly is, can you get rid of the brain? And indeed, we know this happens. In reflexes, this is a very clear thing. The stumbling reflex is a good one. So the image of the little man, your environment changes unexpectedly, your toe catches, and that sensory feedback goes into your spinal cord, which then sends a signal out to lift your leg, pick it up, and avoid falling down, hopefully. And so thinking about this neural circuit and whether this neural circuit can function and exist isolated from the brain is what I like to think about. And what I'm going to do today is tell you a couple of stories. So this is sort of a little bit of data from those collaborations that I've had with Aoki Ishibara and Akio Ishiguro to really discuss and look at what are animals doing in the context of this type of circuit. The way we get at this, I work with intact animals. Well, I'll qualify that, but I work with whole animals. So what I'm interested in is what the outward behavior is of these guys. And if we start with external mechanics, I like to work on species that can move equally well between different environments. And so amphibious animals are a great place to start. So water and land have super different force environments. So if we think about that as one half of a ghost circuit, those environments are going to give a force feedback to the animal that's very different. So whatever they do, however they use their bodies in those environments, is going to change depending on that sensory feedback. So essentially, I'm using environment to change sensory input to see if we can better understand that sensory feedback circuit. I do this also in animals that move fluidly between both environments, so amphibious things. The second thing we do to try to get at this, what's happening at the local circuit, is cutting spinal cords. So if you can remove one part of the system, and we've done this in our lab, numbing lateral lines, taking away vision, seeing how behavior of animals changes. Transecting spinal cords is a really great way of getting the signal from the brain out of the picture. And so using that in combination with different environments, we start to get a picture of how animals are functioning. And then you might say, oh, but if you remove the brain, often animals don't function at all. And this is very true in many mammals, the case. But there are a lot of animals that can actually function without their brains, and I'll show you some examples of that today. But even if, starting off an experiment like this, you lose complete local control because you've severed the brain, it gives you an opportunity to think about the passive internal mechanics of the body. So kind of the other side of the ghost circuit, if you like. What are the actual tissues of the animal able to contribute to the intelligence of the motion? Okay. Oh, I wanted to give you a warning. I always give warnings because I have a student that's very sensitive to centipede pictures. There are going to be some videos of centipedes. They're a little bit heebie-jeebie. So starting, I'm going to talk about work that is really the mastermind of Kotaro Yasui, who is these first videos are when he was a PhD student. He is now a professor and yeah, is doing amazing work. So what I want you to watch in this, you can see on the left here, I've got ground or water. So the top video, the animal is walking. And you can see it does this nice with their legs extended. There's a retrograde wave that goes from head down to tail, and the body is relatively straight. Beautiful locomotion, if you can get over the fact that it's an aggressive, nasty, venomous centipede. The second video here, what you'll see is the animal swimming. So it takes it a little while to get its bearings, recognize that it's in water, but as soon as it does, you'll see it fold its legs and start a large amplitude wave down the body, just like an elongate fish would do. So very interesting. Two different environments, two very different behavioral outcomes. So the question we were wondering is, what is causing that difference in leg use and body use, but leg use in particular? And so the next experiment, we ran centipedes, or Kotaro ran centipedes, moving from land to water and then moving from water to land to get a better, closer look at where is the change happening and is it associated with, truly with, the sensory feedback from the land. So as the animal approaches the water, it realizes getting deep, it's no longer touching the ground, and you can see the legs stop oscillating, the body starts oscillating, and the legs fold in. And if you notice, they do it as they leave the land. So it seems very truly that there's a really specific force feedback sensory input on those legs. As the animal approaches, you can see the legs open, touch the ground, and the animal is walking, moving forward. So if we think of this back to our Brownstone and Bowie idea of what's happening locally in the spinal cord, ground contact is causing leg stepping. And so there's something to do with that sensory feedback of the limb when it touches the ground that causes it to rotate. When there's a lack of ground contact, it appears the legs are folded. So you could say that this sensory feedback is driving that controller to do these two distinct behaviors. The next question really is the animal deciding to do it? Is there top-down brain control that's actually driving this system? Or can we say that the brain is actually not involved and it's truly just the sensory feedback that's allowing these animals to move this way? And so these two videos, top one, the animal is walking, bottom one, the animal is swimming, but these animals have been transected. So what you'll see in the top is really quite amazing. This is an animal that has function in its first half, but is transected for its second half. But you can see as it walks perfectly well. The legs move and it moves forward. He's slowly walking there. They can move actually quite quickly in this state. In swimming, it's a very different story. The front half of the animal is obviously attempting to swim. The legs are folded. The body has an undulation. But the bottom half, post-transection, it's very easy to see. The legs are out in an extended position, and the wave isn't happily passing down the body. So this allows us, just simply by observing, manipulating and observing and using two different environments, to really understand that local sensory input drives limb motion on land, and top-down input drives limb folding in water. So in order to fold and swim, the brain has to be involved. In order to move all those legs in an effective and coordinated way, the brain does not need to be involved. So this pattern or example is really great for showing us that embodied intelligence, or this idea that you can have pretty complex control locally without a brain involved, is very integrated with intention, the brain command to actually behave in a particular way, and that it simplifies control and results actually in maximum performance. So I think this is a really lovely example of how biology is really quite complicated and has many things happening at once. And so we can say, oh, yeah, there's definitely local control, but at the same time, there is also top-down control, and it diversifies and allows biology to be remarkably resilient and versatile in its capacity. Okay, so we learn that environment matters in terms of the sensory force feedback that you get and how it elicits behavior, and that both top-down signals are involved and local circuits without top-down function can be highly functional. So now we're going to move towards another model, which is a fish model. And I'm going to show you a set of experiments that revealed a set of behaviors that these animals can do that tell us something actually quite interesting about internal mechanics. So it's in the American eel. This is a very interesting animal. I'm going to give you a little bit of history of the American eel and thinking about its neurocontrol, because it's a rich history, and I love thinking about science that happened long ago. Anguilliform swimming is really just passing away from nose to tail to move forward. It has a very small amplitude at the start, large amplitude growing towards the tail, and that produces a force and moves the animal forward. Quite beautiful to look at, very soothing while you're doing your PhD. Now, just to map the vertebrate morphology onto this diagram we have of a potential local circuit, higher brain centers pass signals down the spinal cord. This in the bottom left here is a picture of a vertebrate spinal cord. Oops, sorry. Sensory feedback comes in the dorsal ramide there, goes into this bucket of interneurons that is very complex and people are starting to unpack. And then a motor signal comes out to the muscle. So this is how it maps on what we're thinking about. Now, going back in time, in 1934, ages back, a researcher named Holst put forward this reflex chain hypothesis. And so in actual fact, this may have been the first time that embodied intelligence was being talked about in swimming. His idea was that the brain up here sends a signal to the first segment, which causes a muscle contraction. These are in blue are muscles. Muscles are contracting and the body moves. That movement causes a sensory input to the next body segment, which then causes a contraction, which then chain reflex goes all the way down the fish. And so this is the idea. What Holst was thinking is that the brain actually doesn't have to do very much. It sends one signal and locally the animal looks after the rest. This is a very cool idea. So to test this hypothesis, he constrained the motion of the fish, and the idea was that if the fish could not move, you could break that wave and you'd see no wave traveling past that place the animal was constricted. And so this is what he did. So the images are over here. You can see it's quite a setup. I've wrangled eels. Doing this would have been very difficult. What he also did, which was very clever, is that he severed those sensory inputs and motor outputs in the portion of the fish that was constrained, just to make sure that there was no feedback associated with struggling that was reaching the animal. And what he saw was that even though the animal couldn't mechanically pass the wave down its body, there was still activity at the base. And so his conclusion was, oh, actually, the brain is sending a signal all the way to the tail, and that's what's passing the wave. And he concluded that sensory systems are probably not critical for this. So in one moment, he both brought embodied intelligence to the foreground and then said it didn't exist. So, but wait, it's not the end of the world for us. He did inspire Gray, who was another a great scientist at the turn of the last century, to ask this sensory feedback question. Because Gray, was saying, no, there's got to be sensory feedback. And so how does this work? What Gray did was that he transected an eel just behind the head. And what that allowed him to do was to remove any top-down signal from the brain to the rest of the body. And then in these images, you can see he touched the animal at the back end and got a reflexive response. So indeed, that means that there is sensory feedback put back into the system. It exists. And so this opened a whole other round of questions. The other thing that Gray noticed when these fish were transected, although they did not swim, they had subtle rhythmic activity of their bodies, just slightly rhythmic oscillation. And so this was sort of the beginning of the great central pattern generator question, right? CPGs, where did they come from? Gray is starting to see them here. And now I'm not going to talk about any of the history of CPGs, but I am going to jump to 1982. A ton of work was done between Gray and 1982 to really characterize what a CPG was and understand that they existed. The reason I jumped to Wallen is because he has a very wonderful thesis, beautifully done, with a very, very small part of it, with one figure that shows an experiment he ran that's fascinating. And in his experiment, he also transected fish and then listened using EMG to the muscles and saw that post-transection, the fish were actually had a rhythmic signal. And so then he said, well, what if I do 2 transections? So he transected behind the head, he transected mid-body, and he noticed that there were clear signals of muscle contraction and they were coordinated. And that's it in the thesis. And so this really got us engaged and interested in eels and why eels are so special. And so my student Jeffrey decided to take this on and did a series of experiments to revisit what Wallen had done and understand really what's going on. And this is the data I'm going to show you because what he's found are some really interesting things. Very simple experiment, technically kind of challenging, but very easy to understand. You have an eel, you transect it spinally, and you have 4 EMG electrodes. So these yellow dots are places where we had EMG electrodes so we could listen to the muscle above the transection and below the transection. We do it on both sides just in case one of these doesn't work. And we see how active is the fish. And what Jeffrey, and of course, no biomechanics video or talk is good without a deep lab cut video. Jeffrey figured out how to run deep lab cut and managed to digitize all of his videos. So he's got really lovely kinematic data with these animals in different conditions. And these are the 4 conditions that show themselves in his data. Now, each of these conditions has been found they've all been found in a single fish. So we have many fish that we did this with, but it's not that some fish did one thing and other fish did other things. You can see all of these in the same animal. I'll walk you through this a little bit. At the bottom, you can see the eel. The little squiggles are EMG signals, and it just means that the front of the fish is active or the back of the fish is active or both. The red line represents transection. And here we go. There's the fish swimming. You can see that's an intact fish. It is not transected. and it swims in a beautiful anguilliform way. The cord you see is the EMG lead so that we can hear the muscles. The next animal is a transected animal. And you can see in the image below that, of course, it's active at the front of the body because the brain is wanting to swim. It is also active post-transection. So we're finding the same thing that Wallen found, which is great, that these animals do have excitation without brain input, which in itself is really quite impressive because most fish are not like this. The third movie that I'm showing right now is a transected fish with activation at the front half, but no muscle activation at the bottom half. And you can see, quite remarkably, that with no muscle activation, there's really great swimming. So reminiscent of the dead trout, that the body of this animal in that ghost circuit with the fluid is successfully swimming. And then finally, and I think most interestingly, in the last movie, the fish is not swimming with its anterior. So according to the fish, he is just sitting still. Post-transection, however, it spontaneously started beating its tail. So these are active muscles post-transection with no drive coming in from the front. So those are our 4 treatments. I'm going to walk through these now, one at a time, to discuss how this allows us to elucidate some of what might be going on in these local circuits. So an intact fish, you can see the EMGs are beautiful. Anterior, very small magnitude EMGs, but clearly there. As you go towards the tail of the fish, very large EMGs. This is a completely normal pattern for EMG in fishes, in swimming fishes. Body amplitude follows the same lovely pattern that as you start at the head of the fish, 25% body length to 100% body length, your body amplitude increases. So it's just that traveling wave passing along the fish, increasing in amplitude. And then these fish are swimming at about 2 hertz, also a pretty normal speed. So essentially, there's a signal coming down from the top of the head of the fish to power the swimming. If we compare this to a transected fish that has an active EMG post-transection, and so here he is, transection's there, front part of the fish is active, back part of the fish is active, very clearly. Again, really nice EMGs. 2 things to notice. One is that the anterior EMGs are cranked up. So there's more effort being put into that swimming on the non-transected part of the fish, the anterior part of the fish. The result of that is interesting. So you've got the red is now the transected active fish and blue is the intact. So you can see it has the same increase in wave along the body of the fish. And I showed you the videos by eye. It's actually quite hard to see the differences. There is a slight difference here, a slight decline in the amplitude of the tail in the transected animal, but not by much. One thing that's interesting is that the transected animal has a higher frequency. So that brain signal is both higher magnitude and higher frequency. And so what we see there potentially is a sensory imbalance that the fish is feeling. And up in the cerebellum, brain-wise, it's saying, you know, usually when I swim this fast, I should be moving over ground more. And they pick up their speed. So we see that in the EMG, the active EMG at the front of the fish. What we also see is that that sensory feedback, the result of the front of the fish driving the back of the fish, appears to be enough to turn on those local CPGs and have a really nice coupled and coordinated EMG signal. So super nice. If we then take the transected active fish and move it over here and add the transected passive fish, you can see very clearly we've got that increase in amplitude of the EMG at the top of the fish and absolutely nothing downstream of the transection, with 0 change in the kinematic output of the wave. So the body wave just doesn't change. Whether those muscles are active back there or not, you get the same kinematic output, and the frequencies are more or less the same. This variation might be just due to differences in speed of the many trials that we did. So this transected passive fish shows that we've got actually a turning off of that controller. And so that kind of puts into question, what is the role of sensory feedback? Because we've got the same sensory inputs, that body's driving back and forth at the front, but it's not resulting in a regular CPG pattern in the back. Interesting. Finally, the last one I want to show you is comparing the transected spontaneous. So this is the fish that is not swimming at the front end, but the back end spontaneously starts oscillating back and forth. Now, I will tell you that we can get fish to oscillate back and forth if you touch them. So if you put some sensory feedback in there, but they will also spontaneously start doing it all on their own. Whether that's movement of the water that's causing a tiny bit of sensation that starts it, impossible to quantify. But what you see is no EMG activity at the front and large regular activity at the back. Your body amplitude is not that different, a little lower at the front, which you would expect because there's nothing driving. The wave is starting lower back on the fish. And then also what I think is fascinating is that wave frequency is super low. And so this data suggests that controller has a set frequency that it operates at. A lot lower than your regular swimming frequency, but just an innate rhythmic output. Okay, so to summarize this data, you've got your intact fish, there's intention coming down from the top, and it ends up being beautiful anguilliform swimming. No surprise. I want to jump to the spontaneous swimming first, because I think it's the clearest data that says clearly there are CPGs that are active post-transection in this fish. So this means there are groups of neurons that can produce a rhythmic signal without any input from the brain. And that frequency is low. It may or may not be augmented by sensors or by a sensory input at this point. The next one I want to think about is this transection, where you've got active muscle post-transection. So there's no input coming down from the top except for the drive, the mechanical coupling that's going back and forth. What you have here is a controller that we know from this behavior has an innate intrinsic frequency, and you've got the sensory feedback that comes from that anterior portion of the fish driving. And so that sensory feedback is able to coordinate the CPG so that the fish wave from start to finish is really nicely coordinated. Then the last one that's a little harder to understand or explain, really. You've got your anterior swimming, tons of sensory feedback going into that system, but no controller rhythmic output. However, that fish swims kinematically in an identical way to the animal that does have motor output. And so this tells us very clearly that the internal mechanics of the animal, the coupling of the front half of the fish to the bottom half of the fish, is ensuring function. And I love moments like this in biology because it really shows you the redundancy of the system, that without muscles on the back half, you can still swim pretty effectively. That's very useful for, evolution and surviving injury. Okay. The other thing that's very cool in a comparative way between these two animals. Now, remember, evolutionarily, these things are completely divided, right? Centipedes are myriapods. They're like over in the invertebrate section of evolution. Fish are vertebrates. Very, very different. But using this comparative approach, we can say, make some interesting comments about internal mechanics. The centipede tells us things about internal mechanics. And this is, if we remember, the centipede swimming is not so elegant, right? When you take away top-down signaling for the centipede, it loses its capacity to swim. Below is the eel has no muscle activity post-transection, highly effective at swimming. So this is a clear example of how what you're made of matters. Centipedes are stiff, they're crunchy, and without that strong muscle contraction, you're not going to get an oscillation. Whereas an eel is, beautifully supple, and so driving from the front, you can still pass that wave to the tail. Okay, so what the eel tells us is that the brain definitely inputs drive, right? You can have small oscillations or big oscillations coming down as an intention from the top. But when you remove the brain, the sensory feedback and the CPGs can operate independently. What we also see is that potentially those CPGs have the capacity to change their threshold to sensory input so that they can either be on, ready to receive sensory input, or off and ignore it when it's there. And then that tells us also, can they adjust how sensitive they are to that sensory input? And maybe this is what allows these animals to have really diverse locomotor patterns. And really what it tells us is that I really want the mice people to continue their work because they're very close now. There's some really compelling data that suggests they know which interneurons are acting as this comparator. It's complex in there. There's a lot going on, but they need to keep working because I think this is a big black box, and knowing how many interneurons are involved could help us understand how controllers can turn on or off in a similar situation. Okay, so observations. Control of rhythmic activity can exist locally. Great, CPGs as we know them exist and work. Mechanical coupling and body morphology can contribute large amounts of intelligence to a system. So depending on what you're made of, you will respond in a highly effective way, even in a passive system, like the dead trout. The complexity of interneurons in the spinal cord may be able to set different neuron states, so turning that CPG on or off to allow for really diverse behaviors. And then the great thing about biology is that all of these things are running in parallel and provide redundancy that even in the face of injury, catastrophic injury like a spinal transection, you can maintain your performance. All right, and there I will stop and say thank you.
Thank you so much, Emily, for a fascinating talk. Really, 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 really interesting insights, and again, really connects to what we've been doing this morning. Please put any questions in your chat. Just one question I wanted to touch on. You mentioned about evolution and CPGs. I just wanted to dig a little bit further. So is it that process is not really understood, and also what you mentioned about what you're made of matters? So in terms of this controller development, is there any insights into when this happened during the evolutionary process or how?
Good question. I don't have a solid answer for that. Yes, generally neurons don't fossilize. And
Yeah,
so it's very
a bit of a challenge.
And to be honest, I'm not a neurobiologist, so I'm not the one that's using the optogenetics in the mice to actually discover which interneurons are doing what. But talking with my colleagues who do that, it's super exciting and super not understood. So a lot of what's going on with CPGs in biology, we assume function, but then you discover a new interneuron and you realize, oh my gosh, it's working in a slightly different way. Yeah.
Really, really interesting. And I have a few other questions from the chat that I can perhaps paraphrase and try and put to you. One was about the difference in the three types of spinal cuts. Is it just a function, or is it just a functional difference? So is there any size difference or size of damage or type of difference, or just a kind of a functional difference?
Okay, so in our experiment, the data that I showed you, we did do multiple things, but the data that I showed you was all exactly the same type of spinal cut. So we went 44%, I believe, down the body
Yeah.
and severed the cord at that point, and identical procedures across all animals.
Mhm.
So this data is identical across all animals. Yeah. Now, it's a good point because everybody, responds to surgery differently, responds to these sorts of invasive things differently. And so,
Yeah.
yes, it's highly likely that you're going to have some individual variation. That's one of the things we biologists just accept.
Yep.
But at the same time, we follow up with x-rays to see exactly where the cuts are made. You can see the crack in the spinal cord. So you've got
Yep.
to, the way these things put together, you've got to cut through the neural spine to get to the um, spinal cord. And so you can see very clearly that it's completely gone. And then in dissection, of course, too, you can see there's no connectivity there. So in terms of connectivity, we're confident that they're all exactly the same.
Yeah.
How different animals respond, and absolutely every animal has a different intrinsic frequency from
Yep.
my experience. Yeah.
Mhm.
So my CPGs may be running at a different level than your
Interesting.
CPGs.
Yeah.
Yeah.
Made that nicely actually segues into the next question about, do you have any insights into why on land we only need these local control loops while others swimming need the involvement of the brain? Do you think this is related to complexity of the environment, or is it related to which one was kind of the first evolutionary behavior of the animals?
Oh,
Um,
it's a super good question. I do not have a definitive answer. Anything to do with evolution, no definitive
yeah.
answer. However, I think that is a good first hypothesis that these
Yep.
animals evolved most likely. So most centipedes don't swim, just certain species, and it's very specific. And so very much these are animals that evolved to walk and then have gone back. I think you
Yep.
could be safe to say that to swimming. And so then, following that, you could say that the evolution of controlling so many legs, it makes sense that there's a really tight local sensory feedback that helps with that control. Um, and then that would, I mean, it's not a huge leap to think, okay, so that's then why one might suggest you
Yep.
need the brain to switch gaits entirely.
That makes sense. And
Hard to say.
again, yeah, as you say, with evolutionary work, that's very challenging. Again, I think this is another challenging question, but again, coming back to this, what you're made of matters. How do you think there's that coupling between the materials, the structure, the form, and the CPG? Is it frequency, or are there some rules in terms of scaling or body mass? I think we talked about this morning how body mass is such a critical factor. How
Yeah.
does that map to the CPG, its frequency, the number of kind of nodes? Are there any kind of knowns or insights there?
That's a great question too. And I think, I mean, it can answer similarly to how John answered this morning or this early this afternoon. That the smaller you are, the less mass you have,
Hmm.
uh, the faster you can flip,
Yeah.
between states. And so, yes, I think that there's probably a fairly large correlation between the size of the animal and slowness of the
Yep.
animal.
Yes.
Um, yeah, and so yes, it would be really interesting. Eels are fascinating because they come in very small sizes and then they grow to be massive animals. And so you
Mhm.
could actually, it would be technically a huge challenge, but you could run that scaling experiment and
Mm.
see, how do
Yeah.
you how does CPG in just innate CPG frequency change based on size in these animals?
Yeah.
But like philosophically, that's a fascinating question. I would not want to have to use that many animals.
Yes. Yes. And then one final question, again, coming back to the talk we had from John, which was very much based in kind of simulation to work in an evolutionary pipeline. Are there similar tools that you have, or using robot models, as you talked about with Alki and others? I guess that is your two options. So you have either have animal experiments or these kind of robo-physical demonstrators experiments.
Yeah, well, the experiments that I love the most, and I'm only just learning, so
Yeah.
I can't speak to it very greatly, is simulation.
Yeah.
Simulation is where it's at, because you
Okay.
can really manipulate things so finely and get a sense for what are the critical things that can change this system. Now, simulation is based entirely on very simplifications of what's happening in biology. Um, but they do tell us a lot. The 80-20 rule, that you can get a really good distance
Right.
down the road of understanding with a very simple model. And actually Kotaro, who I showed his centipede
Yeah.
work, is working on that and does beautiful things. And I showed you a very